complex piece of IKEA furniture. You get it home, you sit down, crack open the instruction manual, and start building. But there's a typo in the manual. Instead of telling you to use a 1-inch screw, it tells you to use a 10-inch screw, right? So no matter how hard you try to follow the directions, the table ends up totally wobbly, broken, and just completely unusable.
Okay, let's dive into this, because believe it or not, our bodies actually work the exact same way. When the instruction manual of our body has a typo, it results in that wobbly table, which for us means severe illnesses. Now, for most of human history, we just had to live with that broken table. But what if we could go right into the manual, find the exact typo, and rewrite it? Well, that is exactly what a revolutionary technology called CRISPR does. So grab a cup of coffee and let's explore the gene editing miracle that's literally changing medicine forever.
Section 1. DNA and genetic mutations. The broken instruction manual.
To really get CRISPR, we first need to understand what goes wrong in our cellular code. Your genes hold the instructions for making proteins, and those are the essential building blocks that keep your body healthy and functioning. But sometimes there's a change, a typo, in these genetic instructions. Scientists call this a mutation. And these tiny typos matter immensely because they lead to missing proteins or proteins that just don't do their job right. Ultimately, that's what causes severe inherited genetic disorders like sickle cell disease, hemophilia, and cystic fibrosis.
And this brilliantly illustrates just how fragile those biological instructions can be. Imagine you're baking a cake for a friend's birthday, right? And you're following this old recipe passed down from your grandmother. But someone accidentally wrote 1 cup of salt instead of 1 teaspoon. You follow the recipe perfectly, but the cake is absolutely ruined because of just one wrong word. It's the same with us. Your DNA is an instruction manual, and a genetic mutation is really just a spelling mistake on page 42.
Now, what's really interesting about this slide is the staggering scale of the problem. Take sickle cell disease, for example, which is a devastating, painful blood disorder. That entire condition is caused by a single wrong letter or base in the DNA code. Literally just one wrong letter out of the billions of letters in the human body's DNA. It's mind-blowing.
And here's the thing. If people misunderstand this, they might view genetic diseases as this, untreatable fate. But recognizing them as physical typos in our code changes everything. It helps us realize a fundamental truth. If a typo can be written, it can absolutely be erased and fixed.
Section 2. CRISPR-Cas9, the ultimate find and replace tool. So, what exactly is it? Well, CRISPR-Cas9 is a highly precise, programmable technology that allows scientists to remove, add, or alter sections of a DNA sequence. Why does this matter so much? Because before CRISPR, altering genes was incredibly messy and unpredictable. Older methods relied on things like radiation or chemicals that caused random, dangerous breaks in the DNA. CRISPR changes the game entirely. It allows us to actually cure a disease at its genetic root, rather than just treating symptoms for a patient's entire life.
Think about typing a massive 100-page document on your computer. Suddenly, you realize you misspelled your boss's name way back on page 87. Instead of reading every single word to find it, what do? You just use the find and replace function to instantly locate the error and swap it for the correct spelling. That's exactly what this is. CRISPR basically acts like a pair of smart, programmable molecular scissors.
Let's move to and see how this builds, because the origin of this tool is absolutely fascinating. Scientists didn't just invent CRISPR in a lab. Bacteria actually invented it billions of years ago. They use CRISPR as a natural immune system. When a virus attacks, the bacteria snip out a piece of the virus's DNA and archive it. If that same virus ever attacks again, the bacteria use that archived DNA to recognize the threat and chop the returning virus to pieces. Nature is wild, right? Now, if people misunderstand CRISPR, they often fear it's some unnatural, dangerous, sci-fi meddling. But knowing it's actually a natural defense mechanism borrowed right from nature, that helps us see it for what it truly is, a beautiful biological tool, not some scary synthetic monster.
Section 3. How CRISPR actually works. The GPS and the scissors.
Okay, let's break down the mechanics. The CRISPR system is basically made of 2 main parts teaming up. First, you have a piece of RNA called a guide RNA. Second, you have an enzyme protein called Cas9. The guide RNA is programmed to find the specific typo in the DNA, and the Cas9 enzyme follows it to that exact location to cut the DNA strands. Once the cut is made, the cell's natural repair machinery kicks in, allowing scientists to introduce the correct genetic code. And precision here is everything, because if those molecular scissors cut the wrong part of your DNA, what we call an off-target effect, it could accidentally damage a crucial healthy gene.
It's kind of like calling an Uber to get to a very specific address in a massive city. The driver needs the exact coordinates, right? Otherwise, they'll drop you off at the wrong house. In our bodies, the guide RNA is that GPS navigation system, and the Cas9 enzyme is a demolition crew. The GPS guides the crew to the exact broken house. They knock it down, and a sturdy new house can be built in its place.
And get this, scientists have even developed a newer version of this tool called base editing. Base editors are like these tiny molecular machines that can safely rewrite just a single letter of DNA without ever making a complete cut in the DNA strands. So instead of a pair of scissors, it actually works a lot more like a pencil eraser.
If we ignore how precise this mechanism is, we might wrongly assume CRISPR is just out there randomly hacking away at our cells. But understanding that carefully controlled GPS guide proves just how incredibly targeted this therapy really is.
Section 4. The real-world cures and the catch. Delivery and cost.
CRISPR isn't just theory anymore, guys, it's reality. The FDA recently approved the very first CRISPR-based gene therapies, like a drug called Casgevy, treating sickle cell disease and beta thalassemia in patients as young as 2 years old. That's huge. But here's the catch. Getting these CRISPR machines into the exact right organs, targeted delivery, is unbelievably hard. CRISPR works perfectly in a petri dish, but safely delivering it inside a living human body without causing toxicity, that is one of the final holy grails of modern medicine. And on top of that, these therapies pose massive logistical and ethical challenges for healthcare access.
It's kind of like a car company inventing a perfect vehicle that runs completely on water and cleans the environment, but it costs $50 million. The technology is perfect, but the access is totally broken. Physically, delivering CRISPR is like trying to mail a highly fragile package across the world without a protective box. To solve this, scientists are using things like lipid nanoparticles, tiny fat bubbles, or harmless viral vectors to act as delivery trucks to safely transport CRISPR to the right cells.
So, the crucial point is the cost. Casgevy, this absolute miracle CRISPR therapy, comes with a jaw-dropping price tag of approximately $2.2 million per patient. Let that sink in. If we just focus on the miracle cures and ignore these targeted delivery and financial challenges, we're basically going to sit around expecting magic pills tomorrow. But by understanding these hurdles, it empowers us to demand better healthcare policies, push for more research funding, and ensure these life-saving cures actually become accessible to everyone, not just the ultra-wealthy.
Section 5. Practical takeaway. The era of genomic medicine.
So, what should you take away from all this? You should care, because the era of genomic medicine is officially here. We are transitioning from a world where we just manage chronic symptoms to a world where we can actively rewrite the underlying code to prevent suffering entirely. Understanding CRISPR means you aren't just learning biology, you're learning about the future of your own healthcare.
So I want to leave you with a question. The next time you hear about a new gene editing breakthrough on the news, skip all that confusing scientific jargon. Will you picture a molecular GPS and a pair of programmable scissors, finally fixing the typos of life?